when we look at the word biochemistry, we actually see two different words. We've got bio and we've got chemistry. So biochemistry deals with the chemistry of living organisms. And when we talk about the chemistry of living organisms, the compounds in those living organisms contain carbon atoms, hydrogen atoms, and usually oxygen atoms. In addition, they frequently contain nitrogen, sulfur, and phosphorus atoms as well. But the main component of biochemicals is this carbon atom. So let's spend a couple minutes and just review the structure of a carbon atom. So when we look at a carbon atom has 4 valence electrons. And because of that, it can make 4 fairly strong covalent bonds. What that allows carbon atoms to do is to bond with other carbon atoms to make large, complex, diverse organic compounds that contain carbon atoms in long chains or in ringed structures. So if we look down here on the bottom, we've got our general structure for a simple alkane, methane, that shows our carbon atom with the 4 covalent bonds to each hydrogen. And then here we've just got the ball and stick that shows us that it's got a tetrahedral arrangement. Now when we deal with biomolecules, we're dealing with what are called macromolecules. And when we look at that word, it tells us a lot about what kinds of molecules we're looking at, because the word macro means very large. So typically, molecules that are found in living organisms are very large molecules. And there are 4 basic types that we're going to be dealing with. Our first are carbohydrates. Next, we have lipids. Then we have proteins. And finally, we have what we call nucleic acids. And in this particular video, we're just going to talk about carbohydrates. So let's look at the word carbohydrate and kind of break it down, because again, it's got components to it that help us explain what a carbohydrate is composed of. So we have carbon in the name, and we have hydrate in the name. And remember that when we talk about hydrate, we're dealing with water. So when we talk about carbohydrates, they contain carbon, hydrogen, and oxygen. And again, carbon and water, which contains hydrogen and oxygen. And when we look at the ratio of hydrogen to oxygen in water, it's the same as the ratio in carbohydrates. So when we look at carbohydrates, the ratio of hydrogen to oxygen is in a 2-to-1 ratio. When we deal with carbohydrates, their primary function in living organisms is as an energy source. But they're also components of building materials in organisms as well. And when we deal with carbohydrates, we're going to have an identifying suffix with the name. And the suffix for carbohydrates is typically OSE. So if you see the name of an organic compound and it ends in OSE, chances are you're looking at some kind of carbohydrate. Now, carbohydrates are a macromolecule, but when we break them down, they're made up of what we refer to as monosaccharides. The prefix mono means 1, and saccharide means sugar. So monosaccharides are simple sugars. And they're going to be used as building blocks for more complex carbohydrates. A good example of a monosaccharide is glucose. Glucose is a simple sugar. It's a carbohydrate, so it contains carbon, hydrogen, and oxygen. And the ratio of hydrogen to oxygen is in that 2-to-1 ratio. And when we deal with a glucose molecule, it contains 2 functional groups within its structure. It's going to have a hydroxyl group, and it's going to have a carboxylic acid group. So here we have two structures of glucose. We have a straight chain form for glucose, and if we count up the number of carbons and the number of hydrogens and the number of oxygens, we would see that it's got 6 carbons, 12 hydrogens, and 6 oxygens. Glucose can also be found in a ringed form, which would have a similar structure. Now, when we look at monosaccharides, there are several different kinds. Glucose we just looked at. Another one is fructose. And fructose ends in OSE, which tells us it's a carbohydrate. If it's an isomer of glucose, that means that it's going to have the same types of atoms, the same number of each type of atom, but the arrangement is going to be slightly different. And if we remember that glucose is C6H12O6, fructose is going to have the same number of carbon, hydrogen, and oxygens, but they're going to be arranged a little bit differently. So if we look over on the left-hand side, we're looking at the chain structure of glucose. And over on the right-hand side, we're looking at the chain structure for fructose. And really the only difference in the structure is in where the location of that double-bonded oxygen is found on the chain. Now, if we take monosaccharides and bond them together, we're going to make larger macromolecules. 2 monosaccharides are going to make a disaccharide. Okay, the prefix di means 2, and mono means 1, so if we take 2 monosaccharides and bond them together, we're going to make a disaccharide. So a disaccharide is 2 monosaccharides that are bonded together. When we look at them, the general structure we're going to see is C12H22O11. And again, since we're looking at a carbohydrate, the ratio of hydrogen to oxygen is 2:1. Now let's look at a couple of examples of disaccharides, and they may sound familiar to you. Okay, you've got sucrose, lactose, and maltose. They're both all three are carbohydrates. They end in OSE. But they're made from different combinations of monosaccharides. So if we were to look at sucrose, which is a disaccharide, it would be made of a maltose combined with a fructose. And that's table sugar, the stuff you put in your coffee or on your cereal in the morning. If we talk about lactose, again it ends in OSE, it would be a glucose molecule and a monosaccharide called a galactose molecule. And lactose is milk sugar. Someone who's lactose intolerant can't digest that particular disaccharide. And then we've got a third example here, which is called maltose. And maltose is formed by the combination of 2 glucose molecules. So let's look at how this actual combination of 2 monosaccharides to form a disaccharide actually occurs. So here's an example. Okay, here we have a ringed form of glucose. And here we have a ringed form of fructose. And when these 2 simple sugars or monosaccharides combine together, what's going to happen is this hydroxyl group here is going to be removed. And the hydrogen here is going to be removed. And that hydroxyl and hydrogen are going to combine together. And that's going to give us a water molecule. Now when we remove the hydrogen from here, oxygen's going to need something else to bond to. And when we remove the hydroxyl group here, this carbon is going to need something to bond to, so they're going to bond to each other. So we're going to make this bond right here, which links together our glucose and our fructose molecule, and it forms the disaccharide sucrose. And again, we remove that hydroxyl and hydrogen, so we're going to form a water molecule. So this process is called dehydration synthesis. And think about that literally. Dehydration means we're losing water, and synthesis means we're building up. So it's a dehydration synthesis process. So let's look at another example. Let's say we've got a glucose molecule here, and we've got a glucose molecule here, and we're going to combine them together by this process called dehydration synthesis to form the disaccharide maltose. The same process is going to take place that we used to form our sucrose. We're going to remove a hydroxyl group from our glucose and a hydrogen from this glucose. They're going to come together and form water. There's our dehydration part. And then these open spaces are going to come together and form a new bond, which will link our two monosaccharides together and form a disaccharide. And depending on what the monosaccharides are, will determine what the disaccharide becomes. Now, we can continue to add monosaccharides on, and once we start to do that and we get bigger than 2 monosaccharides, we're going to start looking at what we call polysaccharides, which literally means many sugar molecules. So now we're going to see many monosaccharides linked together, and the process is still going to be that dehydration synthesis process. And for each monosaccharide that we add to our polysaccharide chain, we're going to remove 1 water molecule. When we look at polysaccharides, they're classified as starches and cellulose. Starches are things that we can chemically digest. And the reason for that is they have bonds that hold those glucose molecules together, which are called alpha bonds. And we have the enzymes in our body that enable us to break those alpha bonds or digest starches. The other type of polysaccharide are cellulose molecules, which are long chains of monosaccharides, but this time, in addition to having alpha bonds, they have these stronger beta bonds that our body can't digest. So if we look at these structures here, we're looking at starch that's in a potato. And if we look at it, it's nothing but a big long chain of glucose molecules that are bonded together by these alpha bonds that our body can digest. Okay, if we look here, what we're looking at are long chains of glucose that our bodies store in our muscle tissues in the form of glycogen. And again, these alpha bonds that hold these together are things that our body can break down so we can digest this. But if we were to look at cellulose, cellulose contains these beta bonds, which our body does not have the ability to digest. And when we look at these, we typically find them in cell walls, in structures in animals that our bodies can't digest. So let's look at a couple of other examples of polysaccharides. And one would be what we call chitin. And chitin is the major component in the exoskeleton of arthropods. And when we deal with arthropods, arthropods are things like insects, crustaceans like lobsters. They're the things when you step on them, they crunch. And when we look at these chitin molecules, they contain carbon, hydrogen, and oxygen, but they also contain nitrogen, which gives them their strong structure. And we talked a little bit about glycogen, so let's deal with this a little more. Glycogen is basically animal starch. It's the way that animals store long chains of glucose molecules in liver and muscle tissue. And those stored glucose molecules can then be used later on as an energy source for that particular animal.